This is a video for chapter 1.2, the ultrastructure of cells, and also encompassing the parts of prokaryotic and eukaryotic cells. Get your pens and pencils ready. There's going to be a lot of drawing here. So speaking of drawing, let's start out with talking about some general guidelines for drawing for IB. So the size of your drawing should always match the complexity. You don't want to make something that's very complex very tiny, okay? So make sure that you're leaving yourself enough room also so that you can show the correct positioning and proportions of things. This is a pretty good drawing of the digestive system down here, and it's important that you have things in the correct placement and also that you have the correct relative size. So for example, the small intestine has a smaller diameter than the large intestine, and it should be drawn that way. We also don't want to use any sketch lines. Okay, if I'm drawing an outline of a cell, I want to make that a continuous line. What I don't want to do is sketch like this. We don't want that. It's always a good idea to start with pencil and then go over the final draft in pen. Sometimes you realize that you don't want a solid line. So I'm looking at this situation in here maybe, okay, especially when we have overlapping things, it's easier just to start in pencil. Here's the part that sucks about drawing for IB. They never say what they really mean. If you get a prompt that says draw, what it really means is draw and label. Okay, so draw means draw and label. Always include labels. There may be an exception where it says the prompt specifically says you don't have to include labels, but that's extremely rare. So we just want to draw labels like as a common procedure. We also want to draw label lines that are straight. Okay, what we don't want are things like this, things that I tend to do a lot, okay, when I'm just labeling things, drawing things that are curved or drawing things that overlap. We don't want overlapping label lines. This should not look like some kind of interstate bypass junction situation where we have to get a map out to figure where your labels go. They need to be straight lines, okay, that don't intersect. You don't need to draw an arrow either, okay, don't do that. Just simply draw the line. When you're working on an IB exam, okay, you're going to be given a box in which to provide your answers, and the same goes for written answers and drawn diagrams. You always want to stay within the box. Real people grade your IB exams, but they don't look at your actual exam paper. What happens is your paper is going to get scanned in by a computer, and the person who is examining your test is only going to see what was inside the box. So only what's inside the box gets scanned and sent to your examiner. So we want to make sure that whatever we're drawing doesn't actually go outside of the box. Just a couple things to keep in mind. So we're going to talk about a couple of cell types, and of course we're going to draw them. And just in case you're unfamiliar with some of these words, we're going to separate all cells into two broad categories. There are tons of ways I could classify things, but for our purposes, we're going to be talking about prokaryotes, like bacteria, and then eukaryotes. Eukaryotes include animals, plants, fungi, and protists. We're going to spend most of our time talking about plant and animal cells, but there are other types that we kind of accidentally leave out. They all form eukaryotic cells versus bacterial cells that form the prokaryotic domain. And just a little background here about prokaryotes. Okay, they're much smaller and they're not as complex. They're a lot simpler than eukaryotes. So prokaryotic cells smaller, eukaryotic cells bigger with a bunch of stuff in them. They don't enclose their DNA in a nucleus. We get into the bad habit sometimes of saying eukaryotes have a nucleus and prokaryotes don't have a nucleus. And while that's true, that statement can be misleading, okay? Because it tends to make people believe that one has DNA and the other one doesn't. Well, that's not true, okay? Both have DNA. It's just that the prokaryotes don't have a nucleus around it. They also don't have any of these super cool membrane-bound organelles that we'll get to, and bacteria is going to be our primary example. All right, so now it's time to draw one of these suckers. Okay, I'm going to start with the innermost layer, okay, that kind of forms the boundary of the cell, and that is the cell membrane. This cell membrane can also be called the plasma membrane. Same thing, doesn't matter what you call it, okay? Just on the outside of that, all prokaryotes have a cell wall. Okay, so a cell membrane surrounded by a cell wall. All right, what else do they have on the inside? All right, well, they have a big squiggly loop. You're just going to have to trust me that that is a continuous loop. Imagine taking a rubber band that's a loop and squishing it in a bunch of different directions. Okay, and this is the bacterial cell's DNA. Okay, and it is enclosed in what we call a nucleoid region. Okay, so the DNA would be pointing to this actual strand. Okay, the region that where this DNA is found, and I'm going to draw a bracket around this. You don't have to. You could just simply point to that same structure again. That is called the nucleoid region. Again, they don't have a nucleus. Okay, they don't have a membrane around their DNA, but we just call it the nucleoid region. It's where the DNA hangs out. All right, then we've got a bunch of tiny, tiny dots here. Okay, and those tiny dots are ribosomes. Now, notice that I drew a bunch of them. Why did I do that? Well, because bacterial cells have a bunch of them. And so in order to get credit for drawing something like that, you need to understand the relative number that you find in a cell, and we want to make sure that we always show that. Okay, now filling up the rest of the inner part of the cell is the cytoplasm. Okay, the cytoplasm is a water-based liquid. It's got a lot of other stuff dissolved in it, okay, but think of it as kind of like filling up the rest of the cell. Okay, now on some bacterial cells, not all, we're going to find some additional features. These things, all of them have this stuff, okay? Only some of them have the features that we're going to draw next. On the outside of the bacterial cell, it's possible that I may find these little tiny hairy looking things. I promise you they are not hair. Okay, these are something called pili. Pili is plural, okay? The I is how we pluralize things in fancy Latin talk. Okay, a singular one of these would be a pilus, P-I-L-L-U-S. Okay, so pili really refers to all of them. They have a super cool function, which we'll get to next. And then on some bacteria, you're going to find longer structures. Something like that. I'm running out of room here. I'm being very naughty with my drawing. Okay, and these are the flagella. Again, flagella is plural. Okay, a singular extension would be flagellum. So, a couple things to note here. Pili are short, and there are probably much more of them if they're present. Flagella are much longer, and that's because flagella and pili have different structures. So here's a labeled drawing of a prokaryotic cell. On the next few slides, we're going to annotate these structures with their functions. So I hope that you left enough room underneath or next to each one of these to make some notes about their functions. Okay, so here's where we want to start, again, writing down the functions next to each of those structures. Okay, the cell wall is there for protection and for maintaining shape. The plasma membrane, you get a gold star in my book forever if you can remember that plasma membrane and cell membrane mean the same thing, is found just inside of it. And what that does is it controls the movement of materials into and out of the cell. It's kind of like the bouncer of this crazy prokaryotic nightclub, okay? Really regulating what enters and exits the cell. If I'm looking at a micrograph of a prokaryotic cell, really the easiest thing to see is this region in here. Again, that's called the nucleoid region. Okay, so the nucleoid or nucleoid region, okay, is kind of where the free DNA hangs out. We don't want to say that it encloses it because it doesn't wrap around it. It's just an area. Okay, and while we're on this slide, it's important to note that prokaryotes don't have membrane-bound organelles. So all of the cell processes occur in the cytoplasm. So the function of the cytoplasm is that it's the site for cellular processes and reactions. Okay, next up are those crazy pili that I told you about, and they allow bacterial cells to attach to each other and exchange DNA. No joke, these things are so cool. Two bacterial cells can kind of snuggle up together and kind of build one big tube by each attaching both of their pili, and they can exchange little bits of DNA. It's crazy. We're not cool enough to do that, okay? But bacteria are, because they have these structures called pili. Okay, so these little hairy things here are the pili. The flagella, these much longer whip-looking things, are used for movement. So if we think about the tail on a sperm, okay, it's not exactly the same structure, but it has the same function, okay, which is movement, a little swimmer. Okay, next up are the ribosomes. Okay, ribosomes are ubiquitous. We find them in every type of cell. And these are for protein synthesis. So in this cell over here, I can see them as these little purple dots. Notice that there's quite a few. Yikes, in this micrograph over here, it's kind of hard to tell, but you see all these grayish dots in here? Each one of those separate dots is a ribosome. So, one fun fact here, in cells that produce a lot of proteins, like I'm thinking maybe like your liver cells produce a whole lot of proteins for reasons we'll get to later. Okay, those kind of cells have a large number of ribosomes. Okay, so sometimes the number of ribosomes can be an indicator of what kind of function those cells have. Okay, so I hope you left some space near where you labeled the nucleoid region. If not, find somewhere in the margins to jot this down because there's some really important stuff here that we need to know. Okay, first of all, the bacterial chromosome is a single circular loop of DNA. Okay, bacterial cells only have one loop, okay, of DNA. That loop could be all kinds of twisted, okay, but it's still one continuous loop. It is not wrapped around proteins, okay? So some people like to say that it has naked DNA. Yeah, you're probably going to want to be a little more specific than that. There's no proteins there. It's not enclosed in a nucleus, okay? But again, the region that it's found in is called the nucleoid. In addition to this single chromosome, most bacteria also contain what's called a plasmid. Okay, So now would be a pretty good time to go back to your drawing of your prokaryote that you did a minute ago. And in here where you have your nucleoid region with your DNA, separately, I want you to draw a circle of DNA, kind of like you see here. It is a separate loop of DNA. It is not connected to the other chromosome. Okay, and what they do is they carry special genes that have been passed along because they've proven to be advantageous for their environment. Okay, if that's confusing, we can talk more about that later, but for now, let's go ahead and jot that down. And of course, whenever you draw it, you want to label it. So just take a moment to do that now. Okay, now prokaryotes, like bacteria, are unicellular. They're made out of one cell and they reproduce asexually. Now, many of you probably already know that when cells copy themselves and divide, that process is called mitosis. Here, this is a little bit different. This is called binary fission. You don't really need to know too much about it. We just need to know that it's different than mitosis, okay? And that process is called binary fission. Basically, we're going to take that main loop of DNA and we're going to copy it, okay? Those two copies then will attach themselves to different points here, different anchorage points, and that cell splits in half. So it's a little bit different than mitosis, which we talk about with eukaryotic cells. You know, that old prophase, metaphase, anaphase, telophase, all that jazz. Okay, this is just a bit different. This is called binary fission. Bi meaning two, fission, splitting apart, splitting into two. Okay, so before we move on to eukaryotes here, just a quick review. We're talking small cells when we're talking prokaryotes, no nucleus. Okay, the DNA is a single loop and it is not attached to proteins. It has no membrane-bound organelles and they divide by binary fission. And I just realized when I was making this that this is all kind of negative sounding. Small, doesn't have this, doesn't have that, not cool. They do lots of wicked cool stuff. We're just going to get into that later, but we'll come back to that thought. Because now we need to move on to the eukaryotic cells. Okay, so these are the cells that people just love to talk about because they're more interesting. They've got more stuff going on in here. They're bigger, they have a nucleus, they have membrane-bound organelles, okay, and they're compartmentalized, okay, so that reactions can take place at the same time in different places. That's a big deal. That means this increases how efficient our cell is and also what it's capable of. Now, just so you know, what comes along with being pretty and cool and awesome is having to know all of those functions and structure. So just get ready for that. Because there is a long list, friends, of things that you have to be able to identify, draw, and discuss the functions of. Now, since eukaryotes represent several different kingdoms, protists, fungi, plants, and animals, okay, we're going to draw them separately. So we're going to draw an animal cell and we're going to draw a plant cell, but it's important to keep in mind they are both eukaryotes. And we're going to start with an animal cell. You don't need to leave room for annotating functions. I'm going to give you space to go back and do that later. Okay, so animal cells, I generally start with some kind of, I don't know, squishy structure shape, some kind of rounded shape here. I don't know why I'm using red, sorry. Okay, and that of course is the cell membrane or plasma membrane. Okay, and then the other most prominent feature here is the nucleus. Now, this is why I want you to draw in pencil first, but if you only have a pen, that's okay. Just watch how this goes. The nucleus, okay, is kind of round like this, but it's not a solid line. There are tiny little gaps or pores in the nucleus. And that's super important because as we're going to learn in a later chapter, Stuff has to get in and out of there. Okay, so I'm going to go ahead and label a couple of these things here. Okay, this whole general structure is the nucleus. And these holes that I've drawn in that membrane there are called nuclear pores. Okay, so we want to make sure that we have both of those. Inside the nucleus, okay, is going to be a very dark spot like that. That is called the nucleolus. Okay, so we have the nucleus, this whole structure, nuclear pores, the holes, and the nucleolus, a very dark region here, and that's where our ribosomes are going to be produced. What you would find inside the nucleus, it's going to be kind of grayish if you're looking at a micrograph, maybe even looks like it has some swirlies. That's because all of our DNA is hanging out in here. So it's not empty like we're drawing it here. All of the DNA is in here. We're just not going to draw that. We don't need to draw that for here. Now we talked about always putting things in the correct position. Right next to the nucleus is a series of membranes. Okay, so this kind of looks like a stack of pancakes almost. Okay, or maybe there's two of them here. Now, one of these stacks of membranes is going to have little dots on it. Wow, this is really not easy to do with a stylus pen, so I hope you're doing that better than I am. Maybe let's try something else here. Something like this. Oh yeah, okay, super. So some of them are going to have little dots on them. This is a structure called the rough endoplasmic reticulum. Okay, that says reticulum. Sorry. Okay, rough endoplasmic reticulum. And then we have what's called the smooth endoplasmic reticulum. Later on, you can abbreviate this as ER, but when you're drawing and labeling like this, you need to spell out the whole word. Sorry for your life. Okay, so we have the rough and the smooth. The rough has bumps on it, the smooth doesn't. Okay, so we like that. We like when things make sense there. So let's talk about what those dots are. Those are ribosomes. Okay, so these little black things are dots. And ribosomes aren't only found on the rough ER, they're also found in some other places, which we'll get to later. Okay, so we also have some other structures here. Okay, we have another set of membranes that kind of looks like a Wi-Fi signal. Okay, and coming off of them are these little tiny packages, these little droplets. This is called the Golgi. And then you may hear this referred to as the Golgi body, or more commonly the Golgi apparatus. Okay, you have to draw it as a series of little stackable membranes here. And in order to get credit for your drawing, you have to draw these tiny little things here, which are known as vesicles. We're going to learn a lot more about vesicles in a couple chapters, but basically the Golgi packages stuff, and it packages it in what's called a vesicle. So here again, by drawing the vesicles, you're telling the reader that you know the function of the Golgi, and that's good. We like that. Okay, next up is a structure called the mitochondria. Mitochondria kind of looks like a jelly bean with a squiggly something on the inside. We're going to draw that a lot better and more close up later on here. But for now, that's good enough. Mitochondria. And of course, you want to show the relative number, so it's a pretty good idea to go ahead and draw a couple. You don't always have to draw them right next to each other, but that's just working out well for me right now. Okay, next to the nucleus, again, talking about relative size, are these two little tube-shaped things. They often cross over like that, okay? And these are what we call centrioles. They do some cool stuff during cell division. They really kind of look like churros up close, okay? But for now, we're going to leave them like that. It's important that they're near the nucleus, okay? It's also important that the ER is attached to the nucleus as well. It's important that the Golgi is somewhere in between the ER and the cell membrane, okay? You don't have to know why yet, okay? We'll learn that later on in this chapter, but just make sure you have things in the correct relative position. Okay, now we also have these little sacs called lysosomes. Okay, lys meaning to break, some meaning body. Okay, well, maybe that's misleading, but anyways, they're not breaking any bodies per se, but they contain enzymes that help break down old cell parts. They can help break down pathogens. They can help do lots of cool things. And then some animal cells are also going to have other round structures called vacuoles. Okay, I know they look really similar to lysosomes. We can't really help that. The only thing that I'll tell you that can be a good indicator of the difference between lysosomes and vacuoles is that you are much more likely to find more lysosomes, okay, than vacuoles. Vacuoles are generally small in size, few in number, and sometimes absent from cells. Most animal cells have several lysosomes. I don't want to give too much away here, but the more lysosomes we find in a cell, the more likely that cell is found in an organ that's really working hard to produce enzymes. One last thing we're going to add here are some more ribosomes, because you see ribosomes aren't just found on the rough ER, they are found throughout the cell. So we want to make sure that we add that, and then I'm also going to add in that my label points to both type of ribosomes, both free ribosomes floating around in the cytoplasm and ribosomes attached to the rough ER. They both are involved with protein synthesis, but depending on where the protein is going to go, okay, those ribosomes are going to be found in different spots. So if the protein is going to stay in the cell, protein's made in these free ribosomes. If the protein is going to be shipped out of the cell, it's being synthesized here. More on that in another chapter. Okay, next up is the plant cell, where we're going to find a lot of the similar features, just in a different kind of arrangement. So I'm going to start with the outermost layer, and I'm going to draw this as kind of a square. Well, that's supposed to be some kind of square rectangle situation. Okay, some kind of geometric shape that has these angles. That's important. Okay, so this is the outermost layer of a plant cell, which is the cell wall. So kind of like a bacterial or prokaryotic cell. And then it has an inside layer here. And that inside layer is the cell membrane. Okay, now it's important that you put these other structures where they go, because in the end I'm going to rock your world and fill up a bunch of space with one organelle. So let's start with the nucleus. So again, I'm going to draw the nucleus now that I know it has pores. I don't need to go back and erase them. I can just draw my nucleus as a dotted line like that. And it's got a nucleolus in the center. Okay, so again, Here's my nucleus. This is the nuclear pore. Here's the nucleolus. And you know, one thing that I forgot on the last slide, holy crap, make sure you go back and do this on your animal cell. I forgot to label the actual nuclear membrane. Okay, so we've got the nucleus, the whole thing, the nuclear membrane, which goes around the outside of it, the nuclear pore, which is the holes, and then the nucleolus. Attached to the nucleus is our rough ER and our smooth ER. So a series of membranes here. Again, on my rough ER, I'm going to find ribosomes. All right, so what I also need to find is room to label here. Let's see how this goes. Okay, so rough endoplasmic reticulum. That's this. The smooth ER. Is right here. Okay, so now I'm going to fill in my Golgi somewhere where I have room. And again, I want to draw those tiny vesicles, those little tiny sacs. So here's the Golgi. Just saying Golgi is fine, Golgi or Golgi apparatus, but I'm running out of room, sorry. Okay, so we are going to find those here. Plant cells do have mitochondria. Okay, so something like that, mitochondria. Okay, and it might even have some more of them. I don't need to label all of them. As long as I draw them the same, the reader will assume that that means the same structure. Okay, and it also has several organelles that are kind of like a jelly bean shape with some stacks of discs on the inside. Okay, so those are stacks of discs on the inside of these jelly bean things. Okay, and those are the chloroplasts. Okay, so this whole structure here is a chloroplast. We're going to draw that in more detail later. Why am I drawing everything around the edges? Great question. That's because the structure that takes up most of the room in a plant cell, okay, is called the vacuole. And it's storing a bunch of water and it blows up real big, okay? And it kind of squishes these guys out towards the perimeter. So if you look at a plant cell, you should be able to see the cell wall, and then you're going to see a bunch of junk squished up against the edges, and then this big vacuole on the inside. Okay, so I've given you a chart that you can use to help you fill in the functions of the organelles. So let's start with cytoplasm. It's not really an organelle, but it's an important part, okay? So the cytoplasm is really the word for the whole cell, except for two parts, the cell membrane, which is the border, and the nucleus, okay? So it's all this other stuff. Now, this is where the organelles are found. This is also where a lot of the biochemical reactions take place. Sometimes you're going to hear this word cytosol. That just means the fluid part, okay? And not all the organelles and other stuff floating around in the cytoplasm. Now, I don't want you really to write this down yet. I'm going to have a separate box for rough ER and smooth ER, but just so we know, and so we're clear, all ERs, all endoplasmic reticulums, whether they're smooth or rough, are a series of tubes that extend from the nucleus. Okay, so here's the nucleus. They're always going to be found right next to the nucleus, and it's there for transport. Okay, so we're going to talk specifically about what they transport in the next couple slides where we contrast the smooth and the rough. Okay, so what do we need to know about the smooth ER? Well, first of all, it produces, and I would even add, and transports lipids. Okay, for sure. How do I remember that? Well, lipids are things like fats, oils, and waxes. They're smooth, they're slippery, they're slick. Okay, so that's how I remember that. Those lipids can be used for a variety of reasons, and so you may want to jot a couple things down. Those lipids could be used to make the cell membrane, okay, and they could be used to make hormones. Hormones, not all, some, are lipid-based, okay, so all of those lipids are going to be produced by the smooth ER. In contrast, the rough ER, okay, is rough. Okay, we can tell in this picture that here's my nucleus. Okay, here are the rough ER membranes, and I can see those tiny black dots on them. Okay, and those are the ribosomes. So this helps to modify, that means kind of fold things in the correct shape, and transport. So there's that transport word again, not lipids, but proteins. Okay, and so this we also need to know has ribosomes on it. Ribosomes are the site of protein synthesis. So since the rough ER has ribosomes on it, it's a very good assumption that it's making and transporting proteins. How can I tell them apart in pictures or when I'm labeling them? The rough ER is usually closest to the nucleus. So it's usually going to work in a pattern like this, where we have the nucleus, then the rough ER is right next to it, and then the smooth ER would be maybe a little further away. Not always, but sometimes, just kind of a hint when we're looking at confusing pictures like this one. Okay, so let's talk about those ribosomes for a moment. Those ribosomes have no membrane, okay, so they are not an example of what we call a membrane-bound organelle. That's not it, okay? They are found in both prokaryotic and eukaryotic cells. So they are found in all, I don't know what that was supposed to be, all types of cells. Okay, let's not think and write at the same time, Mrs. Hawes. Great, they're found in all cells. Again, these are the sites of protein synthesis. So the rough ER helps modify and transport. Okay, it's the actual ribosomes that are synthesizing or making those proteins. And I can find them in two places. I can find them attached to the rough ER, or I can find them throughout the cytoplasm. There are a couple different types of ribosomes, and we classify them based on their size. Eukaryotes have what are called 80S ribosomes, and prokaryotes have what are called 70S ribosomes. And these have to do with units of size, okay? So the eukaryotes have slightly bigger ribosomes than prokaryotes, 80S and 70S. Okay, now let's take a look at this next organelle called the lysosome. Here's a picture of a liver cell. Again, I know these are really difficult, that's why we're practicing. Here's my nuclear membrane. Here's the nucleolus. Here's all the DNA that's scattered in there. You see all these little black sacs? Many of those are the lysosomes. And here's what we need to remember about lysosomes. Enzymes, 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 enzymes. I cannot say that enough. They contain enzymes, okay? You just have to remember that. Figure out a way. Okay, I usually remember that enzymes can help break things down. Lys means to break. Okay, but if you don't love Latin and Greek as much as me, then find some other way of remembering that. Okay, and those enzymes can be used to break down a lot of different things. Okay, so they can be breaking down particles in our food. They can be breaking down old organelles. Okay, they can break down things that are engulfed by cells. They can be responsible for digestion. There are a lot of things that they can do. I'm not so worried that you know all of those examples right now because we're going to talk about them later. But anytime we need something to be broken down in our cell, okay, so maybe we want to remember that. We want to associate that breaking down process with the enzymes that are found in lysosomes. Okay, now, no offense to your 7th grade teacher. I'm sure they were lovely and wonderful. But I'm hearing a lot of misnomers about what's really going on with these cell organelles. And when I ask people, what does a Golgi do? They're like, it's the postman. No one knows what you're talking about. Okay, so let's start talking like a grown-up here. The Golgi is responsible for collecting and packaging and transporting molecules. Okay, so I can kind of see how you maybe think it's the postman. My postman doesn't help me wrap things up, but whatever. One side of the Golgi is going to be near the rough ER. You don't necessarily need to write that down. I just want to show you how to find it in pictures. So here's my Golgi. It's always going to be somewhere near the rough ER and the cell membrane, and that location is very important to its function. So it's going to receive proteins and it's going to package them in little sacs called vesicles. Okay, we'll talk a lot more about this throughout the year, but here's the ribosomes on the rough ER. They're making proteins. The rough ER is transporting them and modifying them. They hand them off to the Golgi. The Golgi is going to wrap those proteins in a sac called a vesicle. Okay, and that's why we draw them in our drawings. And that vesicle is going to help in the process for getting them out of the cell. Okay, so a lot of shipping and processing going on here, okay, but we need to remember it's receiving, it's packaging, and those packages are called vesicles. All right, so speaking of things that your 7th grade teacher should have never taught you, I also hear a lot of times when I ask students, what's the function of the mitochondria? They all say, the powerhouse of the cell. What does that mean? I have no idea what a powerhouse is. What does that mean? Okay, if you say powerhouse, that gives some wrong impressions about what's actually going on. Here's what we need to know. They are the site for cellular respiration. You should already have a basic understanding of that process, but if not, don't worry. This is IB biology. We go over that in much greater detail later. Okay, so here we're talking about energy transformation. Okay, we're taking the chemical energy in food and we're transforming it into chemical energy in the form of ATP. So I would go ahead and write that down also, where the chemical energy in food is transformed into chemical energy in the form of ATP. Underline, bold, star, whatever, this molecule ATP. If you don't want to know what it is, trust me, you will by the time you're done with this course. Okay. We also know that this product, this process of cellular respiration produces heat, but we don't need to add that in right now because you already know that. One of the reasons why mitochondria are so gosh darn cool is because they have their own DNA and they have their own ribosomes. They also, this is very important too, have a double membrane. So they have one here on the outside, okay, but then they also have a membrane here, okay, on the inside, a double membrane. So it's kind of like a quesarito, this disgusting, lovely invention from Taco Bell, okay? A shell on the outside, another shell on the inside. Same thing here with a mitochondria. We're going to go into more detail about this stuff, about what the folds and stuff are called later on in this presentation. Okay, for now, it's important to know, okay, you can probably figure this out on your own, Cells that need a lot of energy have a lot of mitochondria. You can say the same about all of these cell organelles. If I'm finding a lot of one particular organelle, then I know that that relates back to that cell's function. So if a cell has a lot of Golgis, it's probably processing a lot of things that need to get shipped outside of the cell, like your glandular cells, cells that produce hormones, things like that. If something needs a lot of mitochondria, it must be a cell that requires a lot of energy, like muscles. Has a lot of lysosomes, it must be producing a lot of enzymes, like your liver. Okay, if it has a lot of ribosomes, again, making a lot of proteins. So just think about these things, okay? It's important to know the function of these things because if we're looking at cells that have them in high numbers, you're going to need to know some possible uses or functions of those cells. Okay, next up is our friend the nucleus. Okay, we've talked about this quite a bit, but I'll go over the highlights. It has a porous membrane, pore meaning hole. Okay, so that porous membrane here is going to allow for communication. It doesn't mean that it gets better cell service, okay? That means that molecules can pass from the inside of the membrane out and vice versa, okay? So that's kind of important. It's usually located in the center of the cell, but as we've already discussed with plants, that's really not a possibility. Okay, and then most nuclei have a dark area called the nucleolus, where ribosomes are made. Also, probably a good idea to recognize the actual function of the nucleus, which is that it isolates and contains the DNA. So kind of keeps the DNA separate from the rest of the cell so that none of the crazy shenanigans going on in the cytoplasm affect the very sensitive DNA. All right, next up are the chloroplast. Kind of important to note here that these are only going to be in our algae and our plant cells, okay? And so we're talking things that are primary producers. These are the sites of photosynthesis. Just like the mitochondria, it has a double membrane, so we have another casereto organelle here. It has its own DNA and it has its own ribosomes. Okay, so there are a lot of similar characteristics between chloroplasts and mitochondria. We'll talk about why in another chapter. Inside of the chloroplast, we're going to find these things called thylakoid discs. That's what these little discs are that we see in the stacks. They kind of look like poker chips. Not that you're old enough to understand what that is, but maybe you've seen it in a movie. Okay, and those contain chlorophyll. So these are going to be green. Okay, next up is something called a centrosome. Okay, so centrosomes occur in all cells, and what they do is they help kind of organize the movement of chromosomes during cell division. So they're pretty dormant for most of the cell's life, but right when that cell gets ready to undergo mitosis, they kind of kick into gear and they produce these spindle fibers, which are going to help drag chromosomes where they need to go. We're always going to find them near the nucleus. One important thing to note is that in animal cells, inside the centrosome, are these special features called centrioles. Remember we labeled those? I told you they look like churros. Hello, was I right or what? Okay, those are going to be inside the centrosome, which is just an area that produces the spindle fibers. Okay, and the centrosomes in animal cells are just going to further help with that process. Okay, but they're only going to be active when the cell is dividing so that they can help move those chromosomes. Okay, next up is the vacuole. And the vacuole is really for storing things, particularly water. It can store other things. Okay, there are different vacuoles for different purposes, but most of them store water. In plants, they are often big, huge, ginormous, take a look at this cool model of a plant cell. Okay, here's the nucleus. Here's a big vacuole filled with water. The reason it's so big is so that it can kind of exert pressure on the cell wall and make that cell very rigid. Okay, more on that later. In animal cells, if they're present at all, they're usually small and in small numbers, but sometimes we're not even going to see them in animal cells. Okay, now I've mentioned chromosomes several times. We mentioned them in prokaryotes, how they're a single loop of DNA. Okay, and I said that the centrosomes help move chromosomes around during cell division, but we want to get down and dirty here with what it really means to be a eukaryotic chromosome. So first of all, that number is going to vary with species. Okay, so humans have a total of 46 chromosomes in their cells. Fruit flies only 4. Okay, it doesn't mean that we're better than fruit flies. Well, it may mean that just a little bit. Okay, but anyways, chromosome number is characteristic to species. They are only present during cell division. Okay, wait a minute. Does that mean we don't have DNA? I just told you chromosomes for humans are 46 in number. So wait a minute. How can we not have them all the time? Well, the DNA is always in your cells. It's just not always present in the form of a chromosome. When the cell is not actively dividing, the DNA is in a form called chromatin. So in most of the cell's lifetime, the DNA is going to be hanging out in this kind of loose, unorganized, well, it's slightly organized, but kind of a loose, not coiled form called chromatin. When the cell is getting ready to divide, that chromatin, okay, kind of coils up and becomes super organized and eventually forms this chromosome shape, which is organized chromatin. It's just a lot easier to move around and to make sure that each new cell gets what they need to in terms of DNA than it is trying to move all this loose junk. So I told you have to draw what the association between DNA and proteins for eukaryotic chromosomes. First, I want you to make sure you jot this down. In eukaryotes, okay, DNA is wrapped around proteins, super important, called histones. Okay, so here we have the DNA, and it basically forms these loops. Okay, like this. And in the middle of these loops are histone proteins. So I'm going to label that histone proteins. Okay, and this stringy stuff here is DNA. Okay, so again, that's what's going on here. Okay, DNA is looped around these histone proteins. If it's coiled enough and organized enough, we're going to get this chromosome form. Okay, while we're in like super drawing mode, it's time to take a closer look at the mitochondria. So we drew them earlier, but I want to go back and draw them in greater detail. Okay, so we know that we have an outer membrane. Okay, it's the caseurido organelle. It's got 2 membranes, so we have the outer membrane. Okay, and then our inner membrane, which in here is kind of drawn in this dark red, is all curled up into these loops. Okay, so I usually, just to make my life simple, watch me draw it, kind of draw it like this. Okay, and so it's a continuous loop. All right, it forms a full circle. Okay, but it's all folded. Okay, so that is the inner membrane. Okay, and what we've got here is a space between the outer membrane and the inner membrane. Okay, and listen to how genius this is. This is called the which means between, intermembrane space. The space between the two membranes. Okay, now we have inside the inner membrane this area called the matrix, and it is as ominous as it sounds. Some funky stuff going on in there, which we'll get to when we get to our chapter on cellular respiration. And there's one more thing that we want to label here, and it's kind of hard to tell what it is from a picture. Okay, but better off in this microscope, micrograph drawing here. You see how my curves are kind of big and fat? Well, that's because there's a limit to how well I can draw these things. In real life, they're like long, skinny curves. These would be long and skinny. Okay, and those curves, what they do is they increase the surface area. So instead of just having another membrane like this, by making then this shape, this finger-like infolding shape, it increases the surface area. And we call those bends in the membrane cristae. Okay, cristae. Not a cool name for a baby, but a cool name, okay, for these infoldings in the inner membrane here. All right, now on to the chloroplast. Okay, here's what it looks like in real life. Okay, so not so dissimilar from what it looks like in drawings. Let's make this even worse by trying to do a drawing of our own, shall we? Okay, so we again have the outer membrane. Okay, and this time the inner membrane isn't all squished up and folded into cristae like the mitochondria. It's kind of chilling out just inside of that outer membrane. So the inner membrane is just inside that outer membrane. So really a lot more like a quesarito. Okay, now it's filled with a couple of things here. Okay, we have these discs, and these are called thylakoid discs. So each little pancake looking thing is a thylakoid disc. A whole stack of them is called a granum. Okay, so one disc, thylakoid. Stack of discs, granum. And you're, of course, going to have several granum in a chloroplast. I am not going to draw them all, but it's kind of easy to see that I'm going to find a lot of them here. Now, it's filled up with a jelly-like substance, okay, that takes up all the rest of this space in here, and that is called the stroma. Okay, so now let's take a minute to review what we've talked about. We talked about a lot of stuff. This video was long, and I'm so sorry. Okay, but just to review here, some similarities between prokaryotes and eukaryotes. They do have quite a few. I would be ready to list them. Okay, they both have a plasma membrane or a cell membrane. They both carry out all the functions of life. Mrs. Grin, metabolism, growth, okay, all of those good things. Both of them have DNA. Their chromosomes don't look the same, but their DNA is pretty similar. And they both have ribosomes. Okay, so it's important to know that they're not so totally different. Okay, they do have a lot in common. But of course, there are some major differences also. So you can use a graphic organizer to kind of help you sort out these differences. If you're asked, however, to compare prokaryotes and eukaryotes, you have to make sure that whatever you say for one, you counter with something for the other. So here we go. For instance, for prokaryotes, DNA is in a loop with no proteins. So I would have to say what the DNA is like in eukaryotes and how it's different from prokaryotes to get that mark. So DNA is in a loop with no proteins for eukaryotes. DNA is wrapped around proteins. So we have no proteins over here, proteins over here. DNA is free in the cytoplasm. DNA is closed in a nucleus. Again, talking about the enclosure or lack thereof. Has membrane-bound organelles, mitochondria, chloroplasts, etc. No membrane-bound organelles. Small 70S ribosomes versus big 80S ribosomes. Again, I'm going to keep saying this. Notice how these match up. When I take time to talk about one, you have to counter with the other. And these are small, okay? They are less than 10 micrometers. These are greater than 10 micrometers. So lot smaller here. Okay, now let's get more specific and talk about the two famous examples of eukaryotic cells, which are plants and animals. They look really different, but really they have quite a bit in common. They both have cytoplasm. They both have a cell membrane. They both have ribosomes, both on the ER and floating around in the cytoplasm. They have vacuoles, usually both of them. Okay, they have a centrosome. Okay, remember that's an area next to the nucleus that helps with cell division. They both have Golgi. They both have smooth and rough ERs, and they both have mitochondria. As some of you have told me, don't get it twisted, okay? Plants have mitochondria. They still do cellular respiration. Okay, we'll talk about that later, but it's really important that you remember that in your drawings. Okay, so how are they different? All right, well, plant cells have a cell wall, animal cells don't. Plants have chloroplast, animals don't. Plants have a super big central vacuole. Okay, we drew that in our picture. Animal cells are either small vacuoles or not there at all. Plant cells take extra carbohydrates and they store them as starch. They're very good at that. Plant cells are adapted for that purpose. Animal cells, on the other hand, store their extra carbohydrates as something called glycogen. You may not really know what that is now. You will later, and then you'll be so happy come May when you're reading these notes again to study for your IB exam that you wrote that down. So you're just going to have to trust me for now. Plant cells have no centrioles in that centrosome area, whereas animal cells do. Those are those churro-looking things. Okay, plant cells have a fixed angular shape due to the cell wall. That's the one that I kind of messed up drawing, so hopefully you got that point. And then animal cells have a more rounded shape because there is no cell wall. These are more blobby. Okay, now we've done a lot of drawings. This video is getting really long, so let's make sure we have in our brains, okay, what the outermost layer on any of these cells would be, okay? So this is a really common question we see on IB exams, and you're going to want to make sure that you know it. In bacterial or prokaryotic cells, they're going to have a cell wall made out of something called peptidoglycan, okay? Gly meaning sugar, peptid, protein, so come some kind of sugary protein matrix thing, okay? We can see that over here. Fungi also have a cell wall, but it's made of chitin, so it's a different kind of carbohydrate. Algae and plants both have cell walls made of cellulose. That's another kind of carbohydrate. And animal cells have no cell wall, but they do have something called an extracellular matrix instead, which we'll talk more about in just a second. Now, why is that cell wall really necessary? Well, those cell walls help those organisms do a couple of things. First, they help maintain the shape. Okay, it's a pretty rigid, tough surface. And it also helps regulate water uptake. So we're going to talk a little bit more about this in one of our future chapters. But a lot of times, plants are in a scenario where water is going to be entering their cell pretty continuously. If it were an animal cell with no cell wall, there is the potential there for so much water to enter and then the cell could burst. Okay, that cell wall on plants kind of helps prevent that bursting from happening. So helping to regulate the water uptake. Now, what about our poor friends, the animal cells? They don't have a cell wall. Instead, they have what's called an extracellular matrix. Okay, extra meaning beyond, cellular, the cell. So beyond the cell means outside of the cell. Okay, that part is very important. So the extracellular matrix is outside of the cell. And it's a network of collagen fibers, proteins, and sugars, again, on the outside of the cell, that strengthen the plasma membrane and provide support, okay, and structure. So here's a picture of a cell. Okay, it looks like an osteocyte to me, a bone cell. Okay, and it's kind of stuck in this extracellular matrix. Okay, all this junk surrounding it almost forms like a mesh, like a net, to help stabilize this cell. And that's the end of chapter 1.2. I know that was long, but thanks for hanging in there. We're going to do a lot of practice in class of identifying and drawing these cell parts. It seems like you got it down, but trust me, when you start seeing stuff like this, then you know we have some more work to do here with our identification skills. All right, that'll do it.